Started.
Today, we are very happy to have Dr. Shiv Raj with us. He is speaking to us on a very, very important and interesting topic. He is speaking to us from China. He is doing excellent work in China, and we will enjoy the talk. We'll have a very nice time. Over to Chetna.
डर मुझे भी लगा फासला देखकर, लेकिन मैं बढ़ती गई मंजिल देखकर। खुद ब खुद पहली बाती गई मेरी मंजिल, मेरा हौसला देखकर। A marvelous evening to one and all. This is Chetna. A very, very warm welcome to this absolutely wonderful, beautiful, and auspicious session of sustainable utilization of plant-based bioactive compounds as anti-cancer, cytotoxic, and enzyme inhibitory agents, a drug development perspective. We are really lucky to have this wonderful session today that brings together so many special people like you. It's my privilege to welcome Honorable Dr. Arvind Deshmukh, the President of Microbiologist Society of India, Chandigarh. He is the one who acts as a pillar and strength of this amazing society and provided us platform for this panel discussion. I extend a hearty welcome to Honorable Dr. Swarnjit Singh, the President of Chandigarh Tri-City, Director of SAS, former Head of Environmental Biotech CSIR MTech. A dream doesn't become reality through magic. It takes sweat, determination, and hard work. Yes, it's a belief endorsed by Dr. Swarnjit Singh, who is working extremely hard and making efforts to invite various dignitaries from diverse fields for the spread of knowledge. Dr. Swarnjit Singh has been listed in top 2% global scientists of the world. Dr. Swarnjit Singh was awarded a prize by the DGCSIR for two of his publications that were listed in 70 top publications of the CSIR in its 70th Foundation Day. He has also contributed to the society and masses by spreading science to the remotest corners of the country. He is president of the Microbiologist Society of India and fellow of so many associations. Dr. Swarnjit is chairman biodiversity and chairman MTCC and Gene Bank IDA in India. He is also the member in two WFCC committees. He is the member of Task Force National Biodiversity Authority, Chennai. He has been awarded Loyola Environmental Award. His publications are in high impact factor journals, and the articles are highly cited in the literature and cited approximately 16,000 times. If we talk about his academic qualifications, then he has done his PhD in biochemistry, Regional Research Lab Jorhat, postdoctorate from the state USA, Karlsruhe Institute of Technology, Germany, and Kobe University, Japan. He has also been awarded Lifetime Achievement Award by Shulini University, Solan, Himachal Pradesh, for his outstanding achievements and enormous contribution to the field of microbial technology and environmental biochemistry. On the behalf of Microbiologist Society of India, Chandigarh, I would like to welcome Honorable Dr. Shivraj Hariram. Sir, we welcome you for this amazing session, and we are really lucky to have you us with today. Dr. Shivraj Hariram has done his MSc and PhD, and currently he's doing he's working as an associate professor in laboratory of medicinal plant and food biotechnology, Zhejiang Chinese Medical University. He obtained his MSc in biotechnology in 2004 and PhD in life sciences from SRTM University, India in 2010. He has also worked as KU Brain Pool postdoctoral fellowship from Konkuk University, currently working as associate professor in the Chinese Medical University, China. He has also received RGNF Research Fellowship UGC India in the year 2006 to 2010. He has 12 years of experience in food science, phytochemistry, and biotechnology and nanotechnology. He had more than 105 research publications, 3 patents, and 6 research projects on his credit. He received 15 national and international research. If his expertise in food science, biochemistry, pharmacology, natural products, nanotechnology, and drug discovery, he has completed various major research projects sanctioned in India, Korea, and China. His research is mainly focused on functional food, natural colorants, drug development, nanotechnology, and phytomedicine. So, this is all about Dr. Shivraj Hariram. Let's just not take much and let's proceed towards today's lecture, the wonderful lecture by Dr. Shivraj Hariram on the topic Sustainable Utilization of Plant-Based Bioactive Compounds as Anti-Cancer, Cytotoxic and Enzyme Inhibitory Agents, a Drug Development Perspective. So over to Dr. Shivraj Hariram, sir.
Good afternoon to all of you. Sir, can I start?
Yes, please start.
Yeah. First of all, I'm very thankful to Surajit Singh. Like he's in connection with me from since last 1 year regarding the Microbial Society of India. And you know, our society is doing very great work in every part of India. And I'm also like a member of this society. So I'm very thankful to Professor A.M. Deshmukh sir, who is like living near to my hometown, Osmanabad. So I'm very thankful to all the member of this Microbiologist Society of India for inviting me to present my research work on plant-based compounds as anti-cancer, cytotoxic, and enzyme inhibitory agents. As we know that nowadays people are like moving towards natural plant-based compounds for therapeutic activities, also for cosmetics, for functional food development, because the synthetic drugs or chemically synthesized drug, they have various kind of side effects on human body. So most of the even in all other world like China, Korea, Japan, also in USA, Germany, people they want to develop different kind of medicines originated from plants and even like our organic farming to combat with various diseases and the side effects developed due to the different chemically synthesized drugs. Here, basically, we are working on medicinal plants and also plant-based food products like fruit waste, vegetable waste, and plant explant. And we also developed the hairy root culture to extract bioactive compounds, and also we developed in vitro method to for enhanced production of bioactive compounds by using a simple explant of any plant by developing hairy root culture or adventitious root culture by expressing the gene of interest in Azotobacter rhizogens. We are familiar with that bacteria. So basically, we are working on different biomolecules, different phytochemicals to combat different diseases, which are mainly involved around the enzymes and also like cancer and diabetes. We know that there are different enzymes which are responsible for metabolic disorders like gout, diabetes. So basically, in our research, we are working on gout. The enzyme responsible for is xanthine oxidase, and also some enzymes related to cancer, like different proteins. Enzymes, nothing but the proteins. So we are inhibiting these enzymes by utilizing different plant-based drugs. So we are familiar with the xanthine oxidase, which is responsible for development of gout by depositing the metabolism amino acid into purine and purine into like different uric acids. The uric acid get deposited into the joints and it causes gout. So these are like different enzymes. So basically, we are doing extraction and analysis of these bacterial compounds. So this is like brief summary about my research work. In our lab, we are working on extraction of value-added products for functional food development and drug development by utilizing different plant-based and different plant-based bacteria compounds, and also plant product-based bacteria compounds, and also analysis of plant products as natural drugs and cosmetic ingredients. Also, we utilizing flowers and fruit waste as a source of natural colorants, and development of food-based phytomedicines and nanomedicines, and hero root culture for enhanced production of secondary metabolites. Recently, I got highly cited researcher award from Clarity, also from KU Research, as a KU Research Pioneer Award from Konkuk, and also research Best Researcher Award from Jejin General Medical University. Recently elected as a fellow of Maharashtra Academy of Science. These are citations, and we developed one polyherbal formulation drug therapy for kidney stone, and that is already commercialized in India, and we are working on the animal study, and this is very effective drug therapy. So basically, this is like a synergetic effect to control the kidney stone. These are the funds, research fund allocated by different agency in India, Korea, China. So basically, the basic workflow of my research is simply first selection of herbal extract or medicinal plants or plant-based products, and their quality evaluation by means of botanical, physical, chemical, biological, and animal studies. For drug development, we have to first select the plant and then explant. Before that, we have to check whether their classification, then physical characterization by means of ash value, then weight. Because if we want to target bioactive compounds, the ash value should be minimum, and the bioactive compounds should be high. Even the essential, secondary metabolites content should be high compared to the essential bioactive compounds. Essential biomolecules like lipids, fats, proteins. And then chemical analysis by means of spectroscopic and chromatographic methods like different HPLC, chromatographic methods like HPLC, HPLC, and spectroscopic methods like LCMS, GCMS, NMR, IR. And finally, biological activity evaluation by using microbial culture, animal cell culture, and different models the animal models that is called animal studies. So we are also doing organoleptic study to develop functional food. Basically, we are extracting different flower and fruit waste colors, and these colorants can be utilized as a functional food to develop functional foods. Functional food means addition of natural colorants or the secondary metabolites into the food, and to enhance its flavor, its taste. So this is all called as sensory evaluation or organoleptic evaluation. So next is metabolic analysis. First selection of sample plant, then extraction of targeted and untargeted compounds. Basically, here we are targeting targeted compounds like flavonoids, phenolics, terpenoids, carotenoids. These are important targeted compounds. Then, after preparation of plant extract, we are going for HPLC. This is primary screening of bioactive compounds. HPLC is a thin, high performance thin layer chromatography, and HPLC, we are familiar with HPLC. So after this quantification, we are going for extraction and purification, and after that, we are analyzing their molecular weight and final characterization by means of LCMS and NMR. Once we finalize the extraction characterization, then we are utilizing these extracted bioactive compounds and their nano drug material for analysis of biological activity by using in silico, in vitro, and in vivo models. We are familiar with in silico means by computational methods, or also called as in silico drug modeling, and in vitro by using different assays like in vitro assay. These are very simple colorant assay. We are using different agents, and we are binding with our plant extract. The effect of plant extract we are checking by using in vitro assays different type of dyes. And then finally in vivo by using different animal models like mouse, rabbit, and etc. And basically we are focusing on cytotoxicity, anti-cancer activity, genotoxicity, and enzyme inhibitory activity. As I told, we are also working on stability, sensory evaluation, and in vitro digestion models. If we want to develop functional food, like these things are very important, like stability, sensory evaluation, and in vitro digestion studies. And finally, we are developing drug and functional food for to make commercial market with collaboration with different industries. Already we have good collaboration in China with the different companies, and they also providing different funding for our natural colorant project and also for cosmetic ingredients. So before starting extraction, we have to know the herbal drug development. For herbal drug development, quality evaluation of herbal extract is very important, even for functional food or even for drug development. So basically, as I told, botanical characterization means macroscopic and microscopic characterization. Macroscopic means safe external external features, size, and microscopic means qualitative, quantitative analysis. These are like qualitative, quantitative. By using simple test tube assays, we can detect different like sugars, proteins, carbohydrates, and different bioactive compounds. This is called as botanical characterization, even the plant classification. And next is physical characterization. We have to check the moisture content, extract value, ash value. So these parameters are very important, because if your plant having high moisture content or high ash value or less extract value, then that plant or that particular like sample is not good for extraction of bioactive compounds. So extract value is very important to get the like high amount of bioactive compounds. So that is called as physical characterization. Then we are going for chemical characterization by means of qualitative and quantitative analysis. In qualitative analysis, simply we are analyzing by using, like as I told, in microscopic analysis. In quantitative, we are going for HPLC fingerprinting of secondary metabolites and DNA fingerprinting. Basically, here we are only targeting the bioactive compounds by means of HPLC, and finally different chromatographic methods like HPLC and spectroscopic methods by using NMR, mass, and like UV spectroscopy. The important part is biological activity. In biological activity, we are targeting basically microbe. Here we have to check our plant, like extract, maybe get some microbial contamination. Also antimicrobial activity, toxicological, pharmacological studies, then antagonistic studies, bacterial and fungal, antifungal, antibacterial studies. So these are different biological activities. Basically here we are targeting cancer, anti-cancer study by using different cancer cell line, and using enzymes, the enzymes which are basically involved in metabolic disorders. And finally, organoleptic study. If you want to develop like functional food, we have to check the color, odor, taste, and texture. These are organoleptic or sensory evaluation will be done by using like different sensory instrumentation, like electronic tongue, electronic nose, or even by formulating different subject and questionnaire and giving our sample to the like different like different people to check whether the color is good or odor or taste is good. So basically, we are working on drug development and functional food development. So this is all about like quality evaluation of herbal extract for food and drug development. As I told, we are also working on different fruit and vegetable waste. So it's very important to nowadays there is a huge production of waste by waste, and most of the scientists, they are using waste for production of different like bioenergy production. You can see these are like different fruit peel, pulp, seeds. These are like different waste generated from vegetable and fruits, and we can utilize these waste for phytochemical analysis, biological activity, bioenergy production, recycling and reuse, and agriculture and animal feed. So fruit waste amount, composition, and types. So depending upon that, we can utilize these fruit waste for phytochemical analysis, biological activities, bioenergy production, recycling and reuse, and agriculture and animal feed development. So basically, you can see fruit waste derived nutrapharmaceuticals we are developing by using these peel, pulp, and seed extract from different fruit and vegetable waste. Here, we are analyzing different phytochemicals like bioactive compounds, fluor, biopolymers, and bitter products. Sometimes bitter products are very like useful to develop different kind of drugs. Then their extraction by means of chromatographic and spectroscopic method. Then in vivo analysis in silico, that is called as biological activity, like antioxidant, anti-cancer, antimicrobial, and etc., like anti-inflammatory and enzyme inhibition. As I told, Many scientists, they are working on bioenergy production like biofuels, biochar, biogas, and these are like all this will be performed by means of fermentation, anaerobic digestion, and pyrolysis. And after this, like the remaining waste we can utilize for recycling and reuse. So that is called as functional food development. Dietary, we can utilize that waste as a dietary fiber source, and byproduct development. That is by means of homogenization, processing, and dehydration. And finally, we can utilize the final waste for in agriculture and as an animal feed by means of soil conditioners, by means of green manure production, by means of feed and fodder production, and natural biofertilizer production by direct application, trimming, and trimming processes. So this is all about residual waste, recycling, and reutilization. So as I told, I am working on medicinal plants and the plant-based products like waste products, basically vegetable and fruit waste. So this is plant metabolomics and biological activity. As I told, first we have to select the sample plant, then their extraction. This already I discussed in previous slides, so I will skip this. Simply extraction and biological activity. So now here, as our we are working on different cancer cell lines. So combating cancers is very important by using these bioactive compounds. And in our lab, so we focused mainly on camptothecin, tensionin, rosmarinic acid, catherinine, and cicocaine. Also, the fruit waste bioactive compounds I will discuss one by one in next slides. So combating cancer. So in this, we have to target different pathways. There are different pathways like RS production, breakdown of mitochondrial membrane potential. Then also different proteins are there like BACS-BCL2 proteins, different capsid cascade, then PI3K, AKT. These are different pathways, and we have to analyze these pathways to induce the apoptosis, and also we have to arrest cell cycle target. Basically, we are targeting G2M phase, that is like growth phase second, and mitotic phase and synthetic phase. And this is we have to induce the apoptosis. Apoptosis we are familiar with. This is like cell programmed death of cell. So this is like programmed death of cell. So for that, we are utilizing all these bacteria compounds, and we are targeting all these different parameters. Like this is called as targeting cell cycle and apoptosis, and targeting targeted and enhanced cancer cell death by using these bioactive compounds. So you can see we selected here 3 types of fruit waste like onion waste, apple waste, and citrus waste. We extracted different compounds from different waste. So we are very familiar with onion, apple, and citrus, but we are only utilizing the edible part, the citrus like pulp or juice. Basically, we are using for preparation of juice, and even as a we can utilize as a fruit. For apple, we are also we are using edible part, inner edible part, not the peel. Just we are peeling the apple and we are throwing the apple waste. Even in the juice production industry, in apple industry, just they are throwing the outer cover. Even daily we are utilizing onion in our kitchen, and we are throwing the outer layers like basal trimming, outer skin, and apical trimming. So we collected all these waste like onion waste, outer skin waste, apple pomace, then citrus peel waste, and we extracted different bioactive compounds like quercetin derivatives like quercetin diglucoside, quercetin monoglucoside, and quercetin from onion peel. From apple, we extracted different triterpenic acid like macelic acid, olenic acid, butyric acid, uol, and erythritol. So these 5 triterpenic acids, and these triterpenic acids are very effective agents for cosmetic development. Already many companies, they are utilizing these triterpenic acid to develop emulsion or creams by using these triterpenic acids. And then we're utilizing citrus waste carotenoids. Like carotenoids are very good source of dietary compounds, and they are very useful for our body to combat with even the heart attack, and also they are very good effective anti-cancer agents. After that, we selected different cancer cell line. As I told, HeLa, the colon cancer cell line, colorectal cancer cell line, breast cancer cell line. We selected different cancer cell line, and we studied their effect on by means of like time dependent and dose dependent assay. And you can see this is like anti-cancer effect. The dark square, it shows the dense like cell viability, but after treatment the cell viability is reduced. You can see here also we compared MDCK and HeLa cell line, and the treated if like at 50 micromolar, the treated sample shows there is no any growth of like cancer cell line. So this is all about. And also we studied the in silico enzyme inhibition. We targeted alpha amylase and glucosidase, and we want to check whether the binding affinity or the binding affinity of particular drug modeling we have done by using in silico enzyme inhibition. So this is all about like different waste and their utilization. So I will discuss one by one. Like as I told, we extracted spiroside from onion peel waste. In our previous project, we extracted different quercetin derivatives, but recently we focused on spiroside. This is an water-soluble compound, and for drug development, water-soluble compound is very important. So we extracted this the red onion. Here we utilize red onion compound. In previous slide, we utilize the red onion quercetin derivatives. Here we utilized the spiroside derivative. So, sorry, spiroside. This is also quercetin derivative, but this is water soluble. And we also developed some nanomaterial by using this spiroside. And we studied this spiroside induced apoptosis in early phase. That is very important. You can see the late apoptosis and early apoptosis. Necrosis, there is no any necrosis for this spiroside. And then we also checked protein expression by analyzing different, like we did the Western blot and we analyzed all different protein. So beta-actin is as a control, and we targeted muding. This is anti-apoptotic protein. So we targeted muding protein, and you can see the expression of this protein is reduced as per the as we increase the time of treatment. At 40 to 48 hours, there is no any band of muding. You can see in control there is a band of muding, but in after 40 treatment with spiroside at 48 hours, there is no any band or protein band. So it indicated that the spiroside inhibited the protein expression, mudin, which is an anti-apoptotic protein.
What is ID?
Then CDK2, capsase 3, and capsase 9. These proteins are cascade proteins are very important to check the progress and proliferation of cancer cells. Like BCL2, BID, we have to inhibit these proteins. So you can see there is also reduction of these proteins. Then we checked the cell viability assay. So there is a reduction of, you can see there is a reduction number of cells reduced in after 72 hours. And we calculated the percent inhibition by means of cytotoxic assay by HeLa cells by means of time dependent and dose dependent assay. Also, we targeted different enzymes and we calculated IC50 value like aromatase enzyme, monoxidase A, monoxidase B, and acetylchoesterase. And we found the spiroside is very effective for all these enzymes compared to the standard drugs available in the market. So from this, we can conclude that the spiroside extracted from red onion is will be utilized as a very strong anti-cancer drug against HeLa, colorectal cancer cell line. But for that, we have to in our lab we are doing some synthesis of nanomaterials to increase its solubility and its bioavailability toward the cancer cell and targeted cancer cell activity. And for that, we are also developing some animal models to check. Recently, we published one paper in food chemistry to target these type of animals by developing different rat models. Next, we extracted from apple pomace, we extracted quercetin 3 glucoside. Here also, you can see all the phases like 24 and 48 hours. At 48 hours, the induction of apoptosis is very high. You can see there is early apoptosis, there is high cell death, and late apoptosis also it passed into late apoptosis, but there is no necrosis. So we can say that Q3G induces time-dependent apoptosis in HeLa cells. We also checked dose-dependent cytotoxicity and time-dependent cytotoxicity by using different concentration of these Q3G and providing treatment with different time intervals like 24, 48, and 72. At 72 hour, there is no visible count of HeLa cells after treatment. We also treated cells Q3G. Treated cells were arrested at S phase and G2M phase. As I told, S phase is very important. We have to target cell cycle. Cell cycle means it's very important for cell growth. That is synthetic phase, G0 phase, G1 phase, like growth phase, synthetic phase, and mitotic phase. These are the important phases. 4 phases is one is G0, G1 phase, G2 phase, and M phase. So we have to target these cell cycle. We have to arrest cell cycle by using these bioactive compounds, and we arrested or we stopped the synthesis of DNA at S phase and G2M phase. So mitotic phase and synthetic phase. And we by means of flow cytometric assay, and we also calculated the apoptosis rate. And you can see at 48 hour, the late apoptosis and early apoptosis is higher compared to the 24. In 24 hour, you can see the viable count, control and the sample. The viable count of the cancer cell is very high, but after 48 hour, the viable cell count reduced. So it indicated that the spirochete is acting as a very strong anti-cancer agent, and we are also working on this for animal studies. So next is citrus waste. We utilize different citrus waste basically for extraction of carotenoids. This is a simple protocol. Extraction of madrine is a variety of citrus. We selected madrine peel. We extracted by means of TLC and HPLC and LCMS. And after extraction, we analyzed these carotenoids and we studied the effect of these extracted carotenoids. Basically, we extracted 2 carotenoids, beta-cryptoxanthin. Here you can see beta-cryptoxanthin and neoxanthin. But in this study, we selected only beta-cryptoxanthin. And we studied the effect of beta-cryptoxanthin on BAX, BCL2, P53, Cas3, Cas7, and Cas9. So here, the mRNA expression of profile of anti-apoptotic BCL2 and pro-apoptotic tumor protein P53, BAX, and Cas7, 3, and 9 were studied. And we found by using different concentration of beta-cryptoxanthin for 24 hours. And we found at 10 micromolar, there is a good expression of these important protein and down regulation of BCL2. You can see there is a down regulation of BCL2, and there is overexpression of the important pro-apoptotic protein. These all are pro-apoptotic protein, only BCL2 and BAX. So you can see the BCL2 BAX concentration is very less at 10 micromolar concentration of beta-cryptoxanthin treatment with 10 micromolar beta-cryptoxanthin. So we studied the relative mRNA expression and fold changes by using the beta-cryptoxanthin extracted from citrus. This is characterization of mass spectrum characterization of carotenoids extracted from citrus waste. This is mass spectra of beta-cryptoxanthin, and this is mass spectra of neoxanthin. Then we also studied the effect of beta-cryptoxanthin treatment on cell reduction and accumulation by using HeLa and MDCK. You can see this is MDCK cell line. There is no any significant effect on normal cell line of beta-cryptoxanthin, but there is a good effect of beta-cryptoxanthin on HeLa cell line. So it means that beta-cryptoxanthin having cytotoxic effect on cancer cell instead of the normal cells. We also studied, you can see here we selected MDCK and HeLa cells, cancer cell line, and we treated with different concentration like 50 micromolar, 10 micromolar, 1 micromolar, and 0.1 micromolar. And if we control, this is control, and we compared the effect of this is MDCK normal cell line. There is no any effect on normal cells. You can see the cell number is not reduced but if we increase drug concentration at 50 micromolar, there is a like slight decrease in like normal cell count. But you can see the HeLa at 50 micromolar, there is complete like reduction of cell count or no visibility of HeLa cell count at 50 micromolar. So it indicates that the overall result indicate that the cryptoxanthin could be used as a prospective drug for the chemoprevention of human cervical carcinoma. That is inhibition of HeLa cell line. Basically, HeLa cell lines are human cervical carcinoma cell lines, and with enhanced production of ROS generation and upregulation of BAX and P53 and downregulation of BCL2. So here, enhanced activity of caspase 3, caspase 7, and 9. These caspase proteins are very important to initiate the apoptosis and also cleavage of nuclear DNA. You can see here, we use tunnel assay to check the cleavage of nucleic DNA. And also we studied the effect on cell cycle. So beta-carotenin induces HeLa cell DNA damage, which is detected by tunnel assay. And here, this is anti-cancer and cytotoxic assay for beta-cryptoxanthin. So overall, we can say that the beta-cryptoxanthin extracted from citrus waste could be used as a prospective drug for prevention of human cervical carcinoma. Again, we extracted triterpenic acids from apple pomaceae. Like this is a simple protocol how the apple pomaceae is generated. It will be generated by processing maybe at home by fresh cut slices, sauces, dried apple, canned apple, and in even industry like a preparation of juice, syrup, jam, vinegar. During all this processing, there is a production of like waste, huge production of seeds, peels, and pulp. So total like peel and pulp and seed, it is called as pomaceae. And this pomaceae, the apple pomaceae, we used for extraction of triterpenic acid, and we utilize this triterpenic acid as an antioxidant and enzyme inhibitory agents. All these data already published in different high impact journals, and you can see this is concentration of extracted compounds like methylenic acid, olenic acid. This is HPLC spectra, and we found the high amount of uracilic acid in the apple pomaceae compared to the other triterpenic acids. Recently, we got funding for saffron waste. Like China, also like China is also in topmost country for production of saffron. India, China, Iran. So even the people all over the world, people utilizing saffron. This is very expensive spice and very important for for preparation of different cuisines or different food dishes. But only we are using anther as a saffron product. What about the petals and sepals and the waste, flower waste? So here we selected all this flower waste and we extracted different compounds. As we know that crocin first and crocin second is already known compound from this saffron flower waste. Sorry, saffron flower. But here we first time we extracted the quercetin, thiosulfuroside, and camphorol, thiosporoside. These are like quercetin derivatives. We extracted these all four compounds and we studied the new protective effect of these compounds. Like in previous research, in previous slides, I discussed about the cytotoxicity. Here, important is protective effect, because some drugs are very effective towards the cancer cell line, but along with that, they will kill the normal flora. So we have to combine some drugs which can protect our normal cell and which can enhance the activity of that cancerous drug. Sorry, the drugs which can act as anti-cancer drugs. So for that, we have to check the protective effect. So first, we generated a different ROS, reactive oxygen species, by treating with H2O2. And this saffron extracted compound, they decrease the ROS production in normal cell line. Even there is no any apoptotic, you can see all these pages, there is no any apoptotic effect on normal cell line. Even we studied the genotoxicity by using alum SIPA root growth, and there is no any alteration in anaphase or telophase or metaphase. This is called as a mitotic. We can calculate the mitotic index by using altered phases and non-altered the phases like metaphase, anaphase, telophase. And depending upon that, we can calculate the mitotic index, and we found the saffron waste bioactive compounds, there is no any alteration or genotoxic effect on onion root tip culture. So we developed this onion root tip culture. Also, we studied genotoxicity by using different animal models. So this is like saffron bioactive compounds, maybe like used as a neuroprotective or protective drugs with and which can like increase the anti-cancer activity of different natural products. Then we also extracted bioactive compound from soybean waste. You can see many countries nowadays, they are utilizing soybean for daily in their food products. So there are different companies, they are producing soybean oil, soybean cake, soybean milk. So there are n number of products by like developed by using soybean. So during this processing, there is production of okra, like that is called as soya waste. So during production, there is a generation of okra, and this okra we can utilize for extraction of isoflavones. These are very like promising bioactive compounds. We extracted isoflavones from this soybean waste, and we used these isoflavones against MCF7, breast cancer cell line, HeLa cell line, and MDCK normal cell line. And we studied the cytotoxic effect of okra and extracted isoflavones, and also we studied the enzyme inhibitory activity. And this data is already published in Food Bioscience Journal. So this is a now this part is very important. As I told, if you want to develop some drug like protective and anti-cancer drug, like we are very familiar with mycophenolic acid. Like this is mycophenolic acid. This is extracted from fungus, and this mycophenolic acid is a very strong anti-cancer agent. But the cytochrome 450, it degrades the mycophenolic acid in metabolites. When we introduce this mycophenolic acid into the cell, it degrades into different metabolites, and the metabolism is increased with less viability and less thermodynamic action. So here we combined mycophenolic acid with quercetin by using liposomes nanoparticle, and we inhibited this quercetin inhibited cytochrome 450 activity. So that's why once we inhibit the activity of the cytochrome 450 enzyme, you can see the mycophenolic acid available easily for inhibition of cancer cell line. So here there is like decrease in metabolism with increase in viability and pharmacodynamic action. So like this is like two opposite studies. In single form, mycophenolic acid having a high metabolism, but if we combine with other drugs like quercetin derivatives, the metabolism get reduced with enhanced viability and thermodynamic action. So this we can utilize. We synthesize the like nanomaterial, and we checked the anti-tumor effect. You can see in control the tumor size, like treatment with free quercetin, treatment with mycophenolic acid, and like mycophenolic acids and quercetin, free quercetin and polysaccharide nanomaterial, and finally mycophenolic acid, the liposome nanomaterial, and quercetin. You can see this combinational therapy reduced the tumor size very like significantly. You can check with control this tumor size. Also, accumulation of drug is very important in liver, tumor, in kidney, spleen, different organs. And we found this combinational therapy like very helpful for accumulation of drug. And this combinational therapy, we can say that remove the hurdles of individual drugs and improve the therapeutic efficacy. Also, combination of this lipopolysaccharide nanoparticle posed higher cancer cell cytotoxicity and anti-tumor effect. Also, elevating the free radical induced oxidative stress generated during the course of chronic therapy of mycophenolic acid. These lipopolysaccharide nanoparticles are stable against conversion of drug into free drug and safe administration of both mycophenolic acid and quercetin. So we can conclude that this therapy possesses potential synchronized delivery of co-administration of drugs with increased viability, therapeutic efficacy, and safety. So this already we published in different high-impact journal. Recently, we were also working on utilization of this is a very famous plant widely used in Mongolia and China. We extracted Q3Z from this plant, and we studied the like protein deactivation and gene expression and cytotoxicity, like antiviral activity, anti-influenza activity of this plant bacteria Q3G. Also, we studied the extraction, green extraction method. So we developed subcritical water extraction of ashwagandha. We all are familiar with ashwagandha. That is the medicinal plant widely used in Ayurveda. And we extracted vitaferin, different vithanolides and vitaferin from this ashwagandha. And we studied the same, like using helasaline and different breast cancer line. But the important factor in this extraction is, instead of going for conventional extraction, we used subcritical water extraction. So this is like addition of like different one extra addition of our research in this study. And we are now familiar, like many people afraid about the China, because COVID started in 2019. So we also worked on COVID, COVID, and we published many research papers on COVID-19, how to combat with the different cytokinin load, or also to develop different drugs. So along with that, it's very important to develop enhanced production of bioactive compounds, because simply cutting plant and like collecting explants is not important. So we developed hairy root culture method for enhanced production of bioactive compounds. So just here we are selected explant, and we treated with agrobacterium resigens by already, which is developed by expressing the desired gene, and then infecting this explant by using this agrobacterium resigens. And after that infection, inoculation, and we like after cultivation, we are going for hairy root culture development, and after that we are going for fermentation for extraction of bioactive compounds by means of chromatographic and spectroscopic method. So this is like enhanced production of bioactive compounds. After enhanced production, we are going for nanocarrier nanometal synthesis, and also biometal synthesis for targeted drug delivery. Nanomaterials by using liposomes or like oil phages, even encapsulation method. And biomaterials means using like gene protein, silk, fibrin protein. We are combining different two drugs, like drug first and second, and we are utilizing these drugs to combat the like cancer and anti-inflammatory studies. So this is all about my research, and these are like some opportunities in life sciences for different students like MBBS. If you want to like develop yourself, like after 12th, you can go to MBBS, MD, MS. So this is like general how to like useful for a student. So you can see how an biologist can grow in academic and non-academic. First, getting degree from science, master degree from like in science, then doctorate and postdoctorate. And these are like different areas we can utilize our knowledge, and we can develop ourselves as a researcher, as a entrepreneur, or also we can as a laboratory technician, as a university lecturer. So this is all about the like different, and these are the skills nowadays. Even biotechnologist can develop different bioinstrumentation. So these are like different transferable skills we can develop by using biological. So basically, if you want to move abroad, like USA, Europe, for China, Korea, and Japan, just we have to go for IELTS and TOEIC and TOEFL. These are like a basic requirement to fulfill to get the admission for PhD and postdoc. So IELTS, TOEFL, TOEIC, these exams are very important, and these are like different grades, like academic and general. Listening, these are like different like section: listening, reading, writing, reading and writing. This is all about IELTS and TOEIC and TOEFL course. So opportunities in life science, like life science related to industry has acquired, like recent days, life science industry acquired considerable clout in the national and international arena. National and multinational companies too expanded their acquisition in the biotech market. Even this COVID opened like huge scope for microbiologists and biotechnologists. We can develop different assays, kit, and also different drugs. There are large corporations producing drugs, enzymes, vaccine, cosmetics, and biopharmaceutics. As I told, COVID-19 expanded much more possibilities for research career and job opportunities for life scientists. So these are like life science job opportunities in research, pharma sector, food industry sector, bioprocessing industry, agriculture sector, even setting up your own project and teaching academic sector. We can develop yourself as a medical laboratory, veterinary industry, biotechnology, research and development, then pharmaceutical industry, biomedical engineering, agriculture industry, product manufacturing and production. Like these are like different fellowship in India. We are familiar with CSIR, ICMR, ICR, DBT, UGC, DST, which offer different fellowships. Basically, now I am discussing about fellowships in China and Korea, because I worked in China and Korea more than 12 years. So in China, if you want to pursue your PG or even your PhD and postdoctoral fellowship, they have different fellowships like CAS fellowship, CAS President International Fellowship. In Korea, NRF, KRF, KGSP, and Brainpool Fellowship. In others, various PhD and postdoc scientists, they have their own funding, they can invite. And you can visit this site like Pro Fellowships. You can get all details about the different fellowships. And you can also join our group. Even you can join our microbiologist group in India. So this is all about like in research, you can develop as a research associate, research scientist, scientist associate, scientific director. These are different areas in research area. And these are important like companies in India. I will share this slide to Professor Soranjan Singh, sir, and you can get connected with these industries. So you can see the overview of biotech industry in last from 2018, like 51 billion turnover in 2018. Now almost 5 years past, and you can see how much the turnover is increased. So this is all about like management plant, drug development, their utilization, functional food development. So I'm very thankful to organizing committee, Professor A.M. Deshmukh sir, Professor Soranjan Singh sir, especially thanks to Soranjan Singh sir, and all Microbiology Society of India members and participants. Thank you all. And this is like recently I got highly cited research award, and even I got selected as an editor for different journals. So this is all about like utilization of bacterial compounds from medicinal plants and fruit and vegetable waste. So this is all about my presentation. Thank you. Thank you all.
Thank you very much, Dr. Shivraj. It was an excellent talk. We really enjoyed your talk. It was so wonderful. I learned so many things which I did not know earlier. Can I ask you a couple of questions?
Yes, sir.
Yes. Now, just a couple of questions. So,
yes,
why
sir.
green extraction is important for plant compounds?
Because, as I told, we are working on natural drugs. And if we use conventional methods, like by using some organic or chemically synthesized solvents, like methanol, ethanol. So there is a huge toxic effects on different like agriculture field, even animal cells. So that's why we want to develop green extraction. Those are having very less side effects on even soil, on animal cells. So that's why green extraction methods are very important. Like by using water. Water is universal solvent. If we use universal solvent, water at high temperature, that is called as supercritical, or at optimum temperature, it is called as subcritical. So water is universal solvent, and water, they don't have any side effect or chemical effect on like cell lines. And basically, we are utilizing different cell line. If we extract bioactive compound by using different toxic solvents like methanol, ethanol, then there is no use. So that's why we are going for green extraction method in our research.
Very good. So what are the different techniques used in the study of cancer?
So basically, for cancer research study, if you want to develop in vitro study, we are using SRB assay, MTT assay. These are like different biochemical assay. Here we are target our drug will bind with the like SRB assay, and the concentration of like reduction in drug we can like study by using spectroscopic method. Also flow cytometric analysis, then protein expression by means of Western blot. So these are like different techniques we utilize in cancer study.
Very good. And the last one is, what is the role of functional foods in cancer prevention?
As I told, I worked in Korea and China. like Korean and Chinese people, they are having very proper diet that is called as functional food. They utilize different teas, and that tea contains different bioactive compounds, natural ingredients. So that is called as functional tea or functional food. Even as I told, we are extracting natural colorants, and these natural colorants we are utilizing in different juices. So functional food means having like a specific or control amount of different nutrients and dietary compounds, and we can utilize these dietary compounds or functional food to combat with the cancer. And we also this type of functional food is very useful to combat we will enhance the immunity, because in cancer people there is a decrease in immunity. So if we develop functional food as a drug, we can avoid the decrease in immunity or the effect on normal cell. So that's why functional food development is very important for
Very
cancer.
important. Actually, in Korea and Japan and China, they take a lot of herbs.
Herbs, yeah.
Yes, a lot of herbs like spirulina and all that.
That is called as polyherbal formation, and nowadays it is called as functional foods.
Functional food. Even mushroom is a functional food.
Yes, mushroom also, because mushroom is like now I'm not familiar with India, but in China, in every food they have some mushroom. In every food they have mushroom composition, and mushroom
Actually,
is also a very good source of fiber and flavonoids.
Actually, mushroom is a nutraceutical.
Yes, nutraceutical.
It has a lot of nutrients and salts.
Yes.
So it's a very, very good food.
Yes, sir.
And if anybody has any question, you can ask. Do you have queries, Sheikh? Yes, please ask. Dr. Sheikh, please ask the query. Oh, you're in journey. So you just type your query. Type.
If somebody want my contact or some details, just you can connect with Suranjit Singh sir. He knows because I'm going to Chandigarh soon, so we will have a very good collaboration. So you can connect with Suranjit Singh sir, and we will share all details with you. No problem.
Now let me give you a good news about Dr. Shivraj. Dr. Shivraj is selected for a DBT Institute, NABI, NABI, in Mohali. This is a National Institute of Agriculture. So he will be joining NABI, Mohali, near Chandigarh, very soon. So let us clap for him.
Thank you, sir.
Right. Now, since the disturbance is there in the journey, I will forward his query to you. No problem.
Excuse me, sir.
Yes.
I have one query.
Yes, madam.
Yes.
Very good evening, sir. First of all, congratulations to you.
Thank you, madam.
Hi, sir. My query is I'm also working on bioactive compounds. I'm extracting bioactive compounds from grape or maize.
Yes, madam.
So I want to ask, what will be the scope for it? How can I incorporate further? Suppose I have extracted bioactive compounds. Now, what I will do further? Fortification in what? What's the scope for further study?
So you are extracting bioactive compounds from plant? Which plant?
From grapes, sir. Grape or maize.
Grape or maize. Okay.
Yeah.
And you already classified these compounds, analyzed this compound quantification.
And yeah, sir, I have extracted resveratrol, there's anti-cancerous, various that you told, anti-cancerous, antimicrobial, all I have tested. Means what's the further scope? What can I do further for this? Fortification means how can I make it useful for a country? I want to ask.
Yeah, nowadays fortification is very important. You can add these bacteria compounds with different food products or different milk products, and we can make fortified foods. So first, for grapes, basically, different type of anthocyanins and flavonoids are very important in graptomyces. So after extraction, first you can target your study, whether you are going for as an antimicrobial agent or anti-cancer agent. And depending upon that, for madam, you are focusing on like as an antimicrobial agent or anti-cancer?
So both. I will check the properties all, okay, whether my product is anti-inflammatory, antimicrobial. I will check all the properties using animals cell lines.
Like which cell line? Like HeLa, MDCK?
Sir, it was in vitro. Actually, I forgot I have not carried notebook. It's there
Actually,
written.
basically for like, if you want to develop fortified food product or even drug, first, after extraction, we have to do like in vitro assays, antioxidant, anti-cancer, and by using different cell line, at least 5 cell line, because we don't know whether these extracted bacteria compounds are effective against HeLa or breast cancer cell line. We don't know. So we have to select at least 5 different cancer cell line. And after screening, depending upon the like dose-dependent activity and time-dependent activity, depending upon the IC50 value, we can calculate the IC50 value for all cell line. And after that, depending upon IC50 value, you can select the particular cell line which is inhibited, like significantly inhibited by these bacterial compounds. And if you want to develop functional food, you have to go for organoleptic study or sensory evaluation. So these are like different pathways. One is organoleptic study to develop functional food, and other is like as an anti-cancer or antimicrobial. Even nowadays, people targeting on MDR, multi-drug resistance microorganisms. So if you discuss with me on phone, I will get a clear-cut idea, and I will solve the problem.
Yeah, sir.
Okay.
So where I contact number, sir, from where I should.
Yeah. You can connect by mail, or also you can connect with Professor Sorensen.
Okay, sir. Okay, I will surely contact.
Yeah.
Thank you, sir.
Sure. Okay. If there is no more question, then Chetna,
Yes, sir.
yes. Please give the vote of thanks.
Okay, thank you so much. कि तोड़ना है हर पिंजरा मेरी उड़ान अभी बाकी है तोड़ना है हर पिंजरा मेरी उड़ान अभी बाकी है मंजिलें ज़मीं भर नहीं आसमान अभी बाकी है लहरों का तोड़ना नहीं समंदर की बेबसी बाहर खामोशी सही भीतर कई तूफान अभी बाकी है Once again, a marvelous evening to one and all. Always an end to every event, such as this auspicious interview session. And we are really glad that today Honorable Dr. Shivraj Hariram joined us. A heartfelt thanks to you, sir, for taking out your precious time for this amazing session. There is no limit of ambitions, and that they're not restricted to anyone. One can do all the best possible one is anxious to do. Every ambition must be fulfilled with a great will and sincere efforts for the fulfillment of it. And it has rightly been proved by Honorable Dr. Shivraj Hariram. Now, I would like to express my sincere thanks to Honorable Dr. Arvind Deshmukh, who joined us this platform for the panel discussion and for this amazing interview sessions. I would like to express my sincere thanks to Honorable Dr. Swanjit Singh, who is putting lots and lots of efforts for organizing such great sessions. Last but not the least, I would like to express my sincere thanks to all the people who joined us today for this session and made this session a great success. At last, I would like to end this session with these lines. खोल दे पंख मेरे कहता है परिंदा अभी और उड़ान बाकी है जमीन नहीं है मंजिल मेरी जमीन नहीं है मंजिल मेरी अभी पूरा आसमान बाकी है once again I thank you all for your attention. Thank you so much.
Thank you, sir.
Thank you very much.
Excuse me, sir.
Thank
Yes.
you, Chetna, for the beautiful vote of thanks and for
Excuse me, sir.
conducting the seminar. Thank you, Chetna.
Excuse me, sir.
Yes.
This is Kausar. Sir, actually, a small doubt to Dr. Shivaraj, sir.
Yes, sir. Yes, madam.
Sir, you have told like you have commercialized one product after extracting the bioactive secondary metabolites or bioactive compounds. To commercialize the product, they should be stable, no sir. Like how to make them stable is there?
Yeah, actually,
How to
actually,
formulate them?
I commercialize one polyherbal formulation because if you want to commercialize a pure drug, that is very difficult. Like there are different formalities by government and stability issues and all things. So for that we developed polyherbal formulation for kidney stone, and we commercialize that product as a dietary compounds, and which inhibit the degrade the kidney stone. So that is like different.
Okay, sir. Thank you so much. I'm a research scholar, sir, and I was working on bioactive secondary metabolites for the treatment of cancer.
Yes, and if you want to commercialize your pure compound, it should be
Yes.
like novel or new, and
Yes,
you have
sir.
to go
Yes, sir.
different stages like preclinical study and clinical studies. Okay,
Okay, sir.
and for clinical study, it will take maybe 5 years, 6 years, and you have to collaborate with company. If your drug is novel, then you will get like patent, product patent. There are
Okay.
like two type of patent: product patent and process patent. Okay.
Okay.
So
Okay.
if your drug is novel, then we have to do preclinical studies and clinical studies. Preclinical studies means all the study. Just I discuss with you, like in vitro, in silico, in vivo. These are preclinical studies. And once you get all the results, and if your compound is novel compared to other drugs, if you first time you extracted from the plant or a particular explant, then you can go for clinical study. You can type with different medical institutes, biomedical institutes, and you can there are different phases: phase first, second, third. And in first phase, the number of people is very less, 10 people, and we have to check the side effects. Then stability study is very important, and you can increase the stability by means of different. Nowadays, many researchers, they are utilizing nanomaterial or biomaterials to increase the stability. So this is like very long study.
Okay, sir. Thank you so much, sir.
Thank you.
Sir, can you drop your mail ID in the chat box so that it will be very useful for all of us?
Yeah, definitely, no problem.
Hello, sir. Good evening. Sir, am I audible?
So I'm very thankful to Surajit Singh sir. Due to him, I connected with a very good platform and also good people. So thank you all. Thank you for listening. Sir, can I exit?
Thank you, sir.
Thank you, sir.